Take it away.
Hi, yeah, so I'm Joe Hiltz from Rothamsted in the UK, and this project is funded by the levy boards and the UK government, essentially, who wants to work out how best to regulate mixtures of insecticides. And I'm not going to tell you about mixtures, because obviously when you're doing a mixture you need to work out how to combine two different single insecticides. So we started there and thought about, if you have a single insecticide, how should you best apply a single insecticide in order to reduce the development of resistance in your insect population. So that's the dose rate debate, which is the question: does a high dose of insecticide provide optimal insecticide resistance management? And in the UK at least, the sort of consensus is that if you're going to spray, then you should spray a high dose. So there's frequently calls to reduce insecticide usage, but primarily through less spraying in terms of the number of sprays that you apply. But when you do spray, it should be at a high dose. So that's the question we're going to be considering here. And I should emphasize at the start that the model that I'm using, we're going to be looking at reducing selection for resistance, not controls. Any results here are not about the control of the insect pest, which is a different problem entirely. So there's a good reason to think that a high dose will lead to resistance not developing. And what I've got here is 3 different genotypes: susceptible, heterozygote, and resistant, and their logic dose mortality rates. And you can see if we apply a low dose here, then it will kill about 90%, 95% of the susceptibles, about 5% of the heterozygotes, and essentially none of the resistant homozygotes. Whereas if you apply a high dose of insecticide, then you can kill off essentially all the susceptible homozygotes, most of the heterozygotes, and some of the resistant. And in the high dose scenario, you're essentially only left with resistant homozygotes in your population. And if any immigrating individuals come into your population, they'll recombine in a sexual population producing heterozygotes, which will then be killed off in the next generation. So there's good reason to think that a high dose would be good in this situation. So I should say that this work is building on work which Frank van den Bosch, who was going to be doing this talk, has done on fungicides. And in fungicides, their modelling suggests that increasing dose pretty much always increases selection. And they did a publication, a literature review, to see how the experimental work corresponds to their hypothesis. And in fungicides, they found a total of 19 papers which had addressed this explicitly by tracking the selection over time of a particular fungus, particular resistance allele, and seeing if increasing the dose has no effect on or decreases selection. And in the majority of cases they found, increasing the dose increases the selection for resistance. Now, we did a small literature search into the insecticide literature, and we only found one paper which actually addresses this question, which was that increasing dose increases selection, but I'm pretty sure we can't use that for resistance management strategies. So essentially there's no experimental work on this in insecticide literature, so modelling's what we have to go on. And at the start of the project we were given this list of insects which the people who are funding it would be interested in knowing about. This is a fairly large list, incorporating a lot of different insect lifestyles, life histories. So we had to think about what management strategies would be best for different insects, and can we categorize these insects to find out the most important life traits which would lead to the best management strategies. So, for example, if we just take 2 traits here, the number of generations a year and their reproduction mode, is it possible that an insect which has multiple generations a year and is asexual will be different from an insect which has a single generation a year and is sexual, would they have different strategies which are best at reducing the resistance management? And according to the reasoning that I gave earlier about why high dose is good, they should, because that relies on recombination, and you get recombination in a sexually reproducing organism. So the high dose strategy, according to the traditional reasoning, ought to work for sexuals but not for asexual insects. Yes, so those could be categories. So we made a general insect model where we track the density of larvae and adults through time. Larvae mature into adults, adults give birth to larvae, and within each of those two classes we're tracking 3 different genotypes. So this is really only for target site resistance, not for metabolic resistance, where the copy number is an issue because we only have 3 genotypes. But essentially we track the densities of these 3 genotypes in the larvae and the adults over time, and we can see how resistance builds up by looking at the resistance frequency according to these genotypes. And as I've mentioned earlier, as I hinted at earlier, both the larvae and adults can affect a crop, but in this talk I'm not going to be thinking about how your spray affects the damage done by the insect. And the insecticides affect each of the 3 genotypes, as we've shown previously, according to these probit logic dose lines, where the resistance of the heterozygote is specified by this line, and the dominance of that determines where this line lies. So in a recessive allele, this red line will lie over to the black with the susceptible homozygotes, and with a dominant allele, it will be as resistant as the homozygote resistant individuals. So in our model we can represent all of the insects that I showed previously by changing the parameters. And this graph here just shows that if we spray an insecticide on a susceptible insect population day 50 each year, you can see that the susceptible insect population takes a hit at day 50, and over time that selects for the resistant individuals in the population, which are not, in this case, affected essentially at all by the insecticide spray. So that's what we're doing. We're tracking selection over time according to the amount of dose that we apply each year, and see whether a high dose will increase or decrease the rate at which resistance builds up. And as has previously been found, a high dose can be beneficial. So on the left here, I'm showing the same insect population with 3 different sprays here. And this is the resistance allele frequency on the y-axis, and the number of years since we started the selection on the x-axis. And in this left-hand graph, the full dose here leads to resistance developing fastest, whereas on the right-hand graph here, the full dose has a reasonably slow build-up of resistance. The half dose has a rapid development of resistance, and the 10% dose actually doesn't build up very much at all. And essentially what's happening is we have 2 different trade-offs here. We have a trade-off between 2 different things which are going on. So the first point is that a high dose of insecticide will increase the selection pressure for resistance to develop. So with a high dose of insecticide, the resistance is selected for stronger. And the second point is that the high dose of insecticide will increase the dilution from a susceptible immigrating individuals. And I can demonstrate this by using this graph, which is a graph of an insect density over time through a single year, where here we apply a reasonably small dose of insecticide, and the insect density declines and then increases again once the insecticide has died out. And if we apply a larger dose of insecticide, then the insect density crashes a lot more. And if the immigrating densities are the same, which there's no reason to assume they wouldn't be, then the immigrating individuals make up a much larger portion of the population here, than they would have here. So if the immigrants are susceptible, then they will have diluted the population more and reduced the rate at which resistance develops. Now, the question we want to know is, while that's true, if we reduce our dose, how often is lowering the dose going to increase the selection for resistance after a certain time period? So what I'm showing on this graph is two parameters, essentially, which I've found to be very important. So on the x-axis is the mortality of a susceptible population, going from 80% up to 99%. So that's how much of your in-field insect population is killed by a single dose of your insecticide in a susceptible population. And on the y-axis is dominant. So down here we have a recessive allele where the heterozygote is as susceptible to the insecticide as a susceptible homozygote, and at the top a dominant resistant allele. And what I'm showing on this graph is whether or not lowering your dose from 100% a labeled dose to a labeled dose providing this mortality to 50% of that labeled dose increases or decreases the resistance frequency after 5 years. And you can see that in the majority of this space here, lowering your dose decreases the resistance frequency after 5 years. Lowering your dose from 100% to 50% decreases your resistance frequency. Whereas in this small area down here, lowering your dose increases the resistance frequency. So that's that. And on the next, on the next slide, I introduced two more parameters. Firstly, whether the insect population is reproducing sexually or asexually, and also whether we have a haplodiploid insect here. And on the Y columns, I'm increasing the immigration rate. And the emergence is whether or not these insects emerge from an overwintering population or if they instantaneously appear. So what you can see is that in all three of these recombination methods, sexual, asexual, and if you have haplodiploid insect population, this area exists, where lowering your dose to 50% of what you were applying would increase your resistance frequency. And in all cases, that happens when you have a high immigration rate and a recessive allele and a very effective insecticide at full dose. And also, if you have no immigration and no emergence from your overwintering population, that it will always be the case that lowering your dose will decrease the resistance frequency. So that suggests that the traditional reasoning is not necessarily wrong, but is not in the whole story. So in conclusion, if you're lowering the dose, it's most common that resistance will decrease. Your selection for resistance will decrease. Where resistance does increase is when you have a high immigration rate, a recessive resistance allele, and a very effective insecticide. And again, I need to stress that while this suggests that decreasing your dose would be good for resistance, at least in this talk, I've not considered control. So it's something I'm working on right now. But in the field, you need to make sure that you have enough insecticide applied that you will actually control your insect pest and not damage your yields. Okay, yep.
Okay, we have time for a quick question while we're changing over, because I want to try and allow a bit of time at the end for a discussion. Derek.
If you have a dose that's high enough to give you control, even if it's half the field rate, it is by definition a high dose, is it not? Because you killed everything, or at least you killed as many things as you feel you need to.
Well, a lot of crops have a certain tolerance to the amount of damage that they can take. So as long as you're preventing any damage to your crop, then you'd be fine. But if your crop can survive a 50% reduction in the population, then that would be an adequate control, I would suggest. So I don't think you have to kill everything, but obviously that will be determined on a case-by-case basis.
Okay, thanks very much, Joe. We can come back to this later in the general discussion period. But unfortunately, at this time, for the chemical companies, you can't sell more products to reduce resistance rate evolution.
Yeah.
So we have Michael Renton next. Michael's going to be taking us through a journey that covers invertebrates, weeds, and pathogens, which is terrific.
Thank you. Okay, yes, so I'm going to try and cover weeds, invertebrates, pathogens in one talk, which means it's going to be fairly brief on each one of the examples I give and then bring it together with some perspectives that integrate the three different worlds. Because it's brief, I won't talk much about the modelling methods that I use, but usually what I use is computational simulation modelling, often individual-based, so modelling actual individuals and how they interact to make population behaviour, which allows for more biological detail and realism, but has costs in computation and complexity, but I'll talk a little bit about that at the end as well. So into the world of invertebrates first. So just a couple of examples. This one's actually the most far back in history. Evolution of resistance in stored grain pests, which is a good lead into the next talk. This work was actually done mostly by my student, Mingren here, funded by the CRC, Plant Biosecurity. He looked at lesser grain borer, which is a serious pest of stored grain and faces the challenge of evolution of resistance to phosphine. Mingren was a very prolific student, one kind of student you like. He published lots of papers. Some of the key ones, though, was this one about really whether it was necessary to include the realistic genetics. So David and other people here had worked out that there were two genes and worked out how dominant they were, and that they weren't very dominant, worked out the synergism between the genes. And so we worked, the modelling there was to look at whether that information about the real genetics mattered to the evolutionary dynamics. The answer was yes, it did make a big difference. And the other one here was really management focused, looking at which factors you could actually control that would delay the evolution of resistance to phosphine, and that dosage consistency, so getting a good, high or at least effective dose through the whole silo or storage facility was what was most important to delay or avoid resistance evolution. So the second invertebrates example, red-legged earth mite, which we've heard a bit about already today. So this work's done with the other organisations involved, particularly Owen and Paul and Svet. It's about the spread of resistance. So either in idealised landscapes like this, looking at the effects of different land uses and how that slows or speeds up the spread of resistance through a landscape, or in more realistic landscapes like the one on the left there, just trying to say how we would expect resistance once it's appeared, once it's evolved, to spread through a landscape, and whether different land uses can slow that down or be useful. So that modelling has very simple genetics in it so far, but we also did some preliminary work combining spatial spread with some more detailed genetics there to look at some questions such as, is it better to spray fence lines or leave them as a refuge for susceptible populations, with the idea that if you spray, you reduce the populations but leave perhaps an empty niche for colonisation by the resistant populations that are in the fields, or if you leave the refuge, you increase the populations overall but hopefully dilute the resistant frequencies. And the results, as usual, say that it depends on a lot of things, particularly genetics and fitness penalties. All right, so I'll get on to weeds. So the first example here is soil inversion ploughing, whether occasional moldboard ploughing can delay the evolution of resistance. The idea is that you bury the resistant seeds, perhaps you bring up some susceptible seeds that are down lower in the soil, and the idea is you reset the resistance evolution. So this is just some results here. If you focus on the light blue cyan line, that is a no-till system with an occasional, once every 4 years, strategic moldboard, and the top one shows resistance percentage frequency. The bottom plot shows weed numbers, and you'll see that that strategic moldboard plough is the one that gets resistance slowest and builds up weed numbers slowest. So it shows that it is, theoretically at least, a good strategy for delaying resistance and weed build-up. So full details in that paper that just came out there. So that's with Ken Flower as the other author. And then rotation. So the question here is, what is the best way to rotate pre-emergent soil residual herbicides, particularly so looking at trifluralin, Boxer and Sakura. So we have an example here where there's one herbicide being used quite a lot, but then we have two relatively new ones. Work together with Ari and with Roberto and Steve, and then we have good glasshouse and field data on the genetics, the cross-resistance between these different herbicides and allele frequencies. So we've run a whole lot of simulations there, looking at whether different rotations would be more or less effective. And what we get out there is that actually mixtures would be the best. So if you use mixtures, it's going to suppress the resistance a lot more effectively than any of the simple rotations. And then, of course, there's the economic question of whether the extra cost of full-dose mixtures would be worth it. And then spatial. So the idea here is that weeds and resistance are spatially patchy, much more for weeds than for insects probably, and certainly than for fungal pathogens. But almost all of the weed resistance models have assumed spatial homogeneity, that you've got a big homogeneous population. So the basic question is, does this actually matter? Should we be worrying about spatial variability and patchiness? So this is work being done by my student Gail, who's co-supervised by Steve and Mike at ARI as well. And the initial results that she's getting is that, yeah, it actually really matters, perhaps surprisingly, that if you model the spatial variability, you get significantly slower increase rates in the resistant populations. This is just numbers of resistant weeds in a given field. Because basically, if there's a spatial limitation, that resistance gene needs to spread through space. And for weeds, that's not that easy. The spread is slower than for insects or pathogens. And there's also some preliminary results that seem to indicate that lots of these IWM, integrated weed strategies, are probably more effective than we would think if we didn't consider spatial patchiness, as long as they're applied proactively when resistance genes are relatively rare and spatially dispersed across a field. So that's good news. Okay, so on to pathogens. So this is not about fungicides, but it's about the optimal use of resistant crop varieties across both space and time. So GRDC is funding this, and it's together with DAFWA and others across Australia. That's Remy, who's the postdoc, who's the main person working on this project. So the question there is, can we use different rotation strategies and spatial placement, spatial deployment strategies to avoid or delay the evolution of virulence and then the breakdown of resistance of varieties? So the case study that we're applying it to is blackleg of canola. So yeah, the question is, for example, could I rotate canola every second year with wheat if I use a different cultivar with a different resistance gene? Or could I potentially put canola in 2 years in a row as long as I'm using different cultivars? Or could I even stretch it to having canola even more often as long as I am very careful about which varieties and which resistant genes I use? So that's just temporal rotation. Or then spatially, across a landscape, the green could be wheat and then the other colours could be different cultivars. If I have some spatial strategy across a farm or across a region or across a whole landscape, could I also delay the breakdown of resistance? And so Remy's built a model that has multiple genes in it. It's a polygenic model with different genotypes, different alleles in the strains or different mutations, deletions, corresponding to virulence or avirulence. And basically the genes in the pathogen can unlock the resistance genes in the cultivar. And with the possibility of fitness penalties if you've got extra genes that are not needed. And he's tested a whole lot of rotation strategies, so probably no time to go into detail, but basically just showing that when you test all these different suites of rotation strategies, that under certain conditions, rotating resistant cultivars can greatly delay the evolution of virulence. And so this is all fairly theoretical, but under certain conditions it should be possible to do that. But basically it only works if you have very low initial virulence allele frequencies, so it has to be done proactively. There's no point coming in once you've already got a bit of a problem. And it all works much better, of course, if you've got some fitness penalties to keep things going down as well. And he's completed the prototype spatial model. That framework is done. And so the next bit is to calibrate against some empirical data with Angela and Steve and possibly some people at DAFWA to make sure it's getting some real predictions that will be useful in the actual context. So I think then I'll move into looking forward mode. So obviously I work across the three different worlds of weeds, pests and diseases, and I think having had that opportunity, it lets me see that this diagram is pretty accurate. There's some overlap, at least in terms of a modeler, and there's some areas where they're completely separate, and that in itself helps me and all of us, I think, get some useful insights. So for example, just going back to where Joe was talking about this dose question. So I've worked in that now for quite a while and had conversations with Joe and Frank recently in the UK about it. And I think modelling it is really useful because it lets you identify what it is the key factors that might potentially make the difference between the different organisms. So for example, everyone says low doses for fungicides, that seems to be agreed. Most people say high doses for weeds, that seems to be fairly agreed. And I think they're probably both right, and there's certain reasons, such as the very short generation time in fungicides versus relatively long generation time in weeds. Effective population size is so different. You're selecting on haploids versus selecting on diploids or polyploids for weeds, and dispersal is so different that you're getting different levels of populations coming in from outside the area that's under selection. So it makes sense that you actually get these really different recommendations. And then I was going to put insects up there, but since I knew my talk was just after Joe's, I thought I'd better be careful in case I said something that contradicted him. But there's interesting that the resistance is often recessive, which makes a whole other aspect to the question on dose. So continuing to look forward, I mean, this is where fairly personal opinions, but I think there's a lot of potential for using individual-based modelling in this research because it allows for greater levels of biological realism and detail, and I think some of the assumptions in more traditional modelling approaches may actually miss really important parts of the process, such as the fact that populations really are made up of discrete individuals and they are spatially located, and you can actually get a situation where genes are so rare that you can't get them coming together because individuals can't find each other in a field. So spatially explicit's important because space really matters in that same reason. The fact that we're getting increasing computational power means that it's not a limitation to have complex individual-based spatially explicit models because you can run them on supercomputers and get results in reasonable time still. I'm excited by the fact that there's more data on genetics coming in, that when I was first looking at this, people didn't know much about the genetics, and then working in fields where people can actually say, well, we know it's two genes, and this is how dominant they each are, this is how they interact, they're synergistic to this level. So we can put that into the model because the work that I have done shows that that is crucial. The way that the genes interact is really important. Initial frequencies, everyone knows that that's really important, and usually there's no data on that, and then there's some work being done to try and quantify those better. And that means that modelling can be used for proactive rather than reactive managing. We can actually get in and predict that before we see a resistance problem, well before we see a resistance problem, we can actually be starting to do things to stop resistance evolving in the first place. So models, in this way, I think, can help us understand, manage, and predict. And I suspect that the pattern probably goes in this way. People probably understand that understanding is what we need to do first before we can do much. And I think a lot of modelling in this area is still at this level. And then people probably think we need to be able to predict things before we can manage them. But I think often with this kind of modelling, we can at least get directions right. That if we do this kind of stuff, we don't know how long until we get resistance, but at least we know if we do this, it'll be longer until we get resistance. So yeah, models can be useful in those 3 different ways. Thank you.
So again, only time for one quick question while we change over. Nancy.
So, Michael, thanks for your talk. I liked your last slide where you talked about understanding and managing and predicting. I'm quite interested in the idea of can you use models to adapt, so incorporate resistance monitoring data and then try to adapt a management plan. Do you have any thoughts about that? Or, it's not quite the prediction part. I'm just curious if you have some thoughts on that.
So that as data comes in, you actually update the model. Yeah, so I guess we've done that for other kinds of models, but not for this resistance evolution modelling. But yeah, I think that would be a really useful way to approach, yeah, for sure.
For what you've done, your example, for other models, can you...
So for biological incursion kind of modelling, we've done that where you have some estimate of what an incursion is, and then as data comes in, you start to be able to better estimate what the incursion is actually. Yeah.
Okay, thanks, Michael. Okay, so next up we are going to change tack a little bit. So Holly Bear and David Chapelius are going to talk to us about stored product pest resistance management and how those can help. Of course, don't forget that Michael did mention models that were applicable to this situation. So take it away, David. Talk, sir.
Thanks, Ari. We really appreciate the chance to come and give this talk. I'm going to lead off and provide some context, and then I'll hand over to David. Disappeared. Oh, disappeared. I'll hand over to David, and he'll carry on with the monitoring and management. And this project is a long-term collaboration with the Department of Agriculture and the University of Queensland. It started when GRDC funded David on a PhD scholarship, and it's been a brilliant collaboration ever since. So to give you some background about phosphine, we do pest control in a box. So we have a closed system. So in some ways, it requires some different thinking than you'd have in a field situation. But phosphine itself is a remarkably simple molecule. It's just a phosphorus and 3 hydrogens. It's the major way to control pests worldwide. It is highly effective against all species and growth stages of the insect. It's inexpensive, distributes to the grain quite readily, but it's a metabolic toxin, a different type of metabolic than people have been talking about. You've been talking about metabolizing pesticides, breaking them down. When we talk about it, we're talking about a toxin that disrupts energy metabolism. And so it has a slow effect. So it is slow acting. It requires that we have good structures that are well sealed, but it's helped us out in the area of dealing with resistance. So the Department of Agriculture has been monitoring resistance very tightly over the years, and when a higher level resistance comes, then we increase the dose to completely control that. It doesn't leave residues on the grain, and so that's an acceptable means of control. And we can also not only increase the concentration, but also the time of exposure. And that's been useful up until more recently. So phosphine is the only fumigant that's really available for routine fumigation. There's a couple of others that have very restricted use. The major grain pests have strong levels of resistance now. Very recently, Cryptolestes ferrugineus has pushed the very limits of phosphine to control the pest. So we've looked at the genetics of resistance in a fair number of species now, and we find that in every case there are two resistance genes, major resistance genes, resistance to phosphine 1 and resistance to phosphine 2, creative. And weak resistance comes about from that RPH1 gene, and the resistance, depending on the species, is up to about 25 times the basal level of susceptibility. So we have weak resistance here. When we combine RPH1 and RPH2, or when that is combined in the insects, you have resistance alleles of both, then we get very strong synergistic interaction and very high levels of resistance. Now this is a log scale along the bottom, so well and truly it's a multiplicative effect. We have recently determined, not yet published, that RPH1 is a fatty acid desaturase, and that results in the polyunsaturated lipids in the membranes, and pretty much any mutation that will knock out that gene function will give resistance, and as a result, it's easy to get resistance from that. That was the first resistance to arise, and it is now essentially ubiquitous, found throughout grain-growing regions. RPH2 is a different story. RPH2 is the dihydrolipoamide dehydrogenase gene, or DLD for short, and that is an enzyme that is involved in core energy metabolism, so it is that link to that energy metabolism. Being so centrally important, any mutation that knocks out gene function is lethal. And so that won't do. And so there's very specific point mutations in this gene that are required to achieve resistance. Since RPH1 is ubiquitous, when we go to monitor the spread of strong resistance, we just have to monitor those few resistance alleles in RPH2. Now, to just provide a bit more context, how we think this works together, aerobic respiration is essential for phosphine toxicity. Under very low oxygen, phosphine isn't a toxin. So that's essential to trigger the toxicity, and that's where RPH2 is situated, that resistance. And then we get reactive oxygen species produced that interact with unsaturated lipids, triggering a free radical chain reaction, membrane damage. And some of the fatty acids, the damaged fatty acids that are released from that, can act on the energy metabolism enzymes and trigger the further generation of reactive oxygen species. And so it's a self-amplifying system. So that's where I'm going to hand over to Dave, and he'll talk more about the management issues.
Thanks, Paul. Yeah, so Paul gave you a nice brief overview of the genetics underlying phosphine resistance. But when we take it to see what's happening in the field, we found that resistance is the same everywhere. So we looked at a number of different species now. Some of them are unpublished just yet, but we found that the two-gene system, and it's the same two genes, are the same in these four species, red flower beetle, lesser grain borer, rusty grain beetle, and rice weevil. In all of in the countries that we've looked at, we've had collaborators in India, we've had strains from China and Vietnam, Turkey, America, and we find that, so whatever we're talking about phosphine resistance globally, we're really talking about the same thing everywhere. So we don't have these metabolic genes that might pop up in one species or one strain in one country and something else in another. We're really talking about the same thing everywhere. And sort of to bring it back to a bit of the modelling, a bit of the theory about resistance development, there are several factors that increase the selection pressure in a stored grain system. And one is that Australia has a nil tolerance to live insects, and not all countries do. Basically, if you can find a live insect in a parcel of grain, the industry considers that grain unfit to sell because they market it as insect-free. So that applies a massive selection pressure, really, about trying to maintain that that grain without insects. So the economic threshold is set very, very low. And that, of course, pushes on to multiple dosing, which increases the risk of selection, especially of strong resistance. And the longer you store grain, the more you have to dose it to maintain it as insect-free. And also underdosing. And underdosing can happen through several ways. Improperly sealed storages. Some of the storages age, so we have to maintain pressure tests as trying to maintain the dose rates. And sometimes there are deliberately low doses or ad hoc fumigations, which are definitely not recommended, and they're not part of the industry best practice, but sometimes it happens if somebody's under pressure to try and sell their grain and they just don't want a live insect being sampled out of it. So there are also factors that are intrinsic to the system that have delayed resistance development. Phosphine has been used as a fumigant for more than 50 years, and we're only really starting to get higher levels of strong resistance to it, which is really quite different to most of the insecticides that you'd be used to. Once you start seeing a strong resistance, it takes over fairly quickly, or it becomes economically unviable or not useful at all. So, but phosphine, we'd first detected strong resistance about in 1997 in the lesser grain borer, but phosphine is still effective against lesser grain borer. So, and there are a number of factors that contribute to that, and that one is that it is a controlled environment, so it really does facilitate a high dose strategy. So you can maintain doses for higher doses for a longer time, and you can kill resistant insects or reduce their population down below the threshold that you're even able to sample them. And there's no residual effect of phosphine. So there's no environmental or health effects that you're going to get from phosphine. So that's really helped in Australia, that we have actually, when they started detecting strong resistance, a high-dose strategy was recommended and implemented. And you'll see the effects of how that has happened a couple of slides later. But also the two-gene model, so the requirement for the two genes, as Michael has shown you, that modelling has shown that has a really large effect on the fixation of resistance. So if you have, combined with that refuge effect outside of the silo, doesn't get dosed. There's no overspray effect. And so you generally have populations that don't get selected outside, and so you have movement or reinfestation of that grain, and then you would have a dilution of effect of the resistance genes with susceptible insects from outside of the silo. But with a two-gene system, that's almost amplifies that dilution effect. So if you only have a heterozygote of one of those genes, your resistance levels just drop dramatically. And so the GRDC initiated a national resistance monitoring program, and it's probably one of the longest resistance monitoring programs that I think I've encountered. It's been going for more than 25 years, and it was really implemented to provide an early warning sort of system to diagnose failures, to see if the failure was due to resistance or was it due to bad fumigations, leaky silos, or something like that, and give a strategic picture of what was going on. So when they first detected strong resistance, the high-dose strategy was implemented. But it's a phenotype-only assay that was developed because we had no knowledge of the genetic back then, and it requires live insects. So that really limits the sorts of samples that you can get. It's basically where you can find the insects. So usually you can only sample from storages themselves. But out of that program, a number of strains were collected, and we found that there's actually a diversity of resistance alleles in RPH1, but especially also in RPH2 from around Australia. So we had half a dozen different strains of Rhizopertha, and we found about 5 different alleles of RPH2 in those strains from just different parts of the country. So, so it was independently arising from different parts of Australia. So really, it's locally arising, it's locally selected, and they're quite independent. And so we developed a genotyping assay. It's genotyping by sequencing using the Illumina platform. so that we can detect all of those alleles at the one time from several species now. Or we've got two that are working quite well. But it's precise, it can give gene frequencies, and it can detect resistance alleles at very low frequency. So you can detect one resistance allele in 100 insects sampled, which you couldn't do by a phenotype test. So basically, we can detect the resistance before a phenotype, long before a phenotype test will come up with a positive result. And it doesn't require live insects, so it enables trap catchers to be surveyed. So you can sample insects from pheromone traps from outside of a silo, or even away from the silos out in the field. And so, from one of our surveys, we've shown that resistance is fairly diverse in Australia, even though there are a couple of major alleles. We've mostly done the northern region, but in the southern region there's also a couple of local alleles that we haven't really surveyed for yet. But in the northern region there was an overlapping distribution of two major alleles, and one was pretty much confined to Queensland, and the other one, the P49S, that's actually a major allele that we find globally, but the sequence showed that it was arising from Australia, not arising from any of the other countries that we'd sampled. And we can also find that, so we've also found that the proportion of insects carrying resistance, there was a rather large storage type effect. Not for most of these, like shed, silos and unsealed storages, but for storages like bunkers. And we know that bunkers sometimes have, because there's under a tarp, and so that there's always technical issues arising from maintaining doses in those sorts of storages. They leak a bit more than a solid storage. They get attacked by birds, all sorts of things. So, but the surprising thing actually for me is, even though we found the strong resistance arose 20 years ago in Australia, we detected it nearly 20 years ago, the average frequency across the whole region was still less than 10% of insects. And there was only a couple of outliers that had very high frequencies around a central storage that only really had large numbers of bunkers. And there were quite a number of central storages that had no detectable resistance alleles there. And we've taken our genetic information, we've got collaborators in India, and they've never monitored for phosphine resistance. They've only just started becoming interested in it because they're having lots of fumigation failures and difficulty. And they have a much different system. So they fumigate in bag stacks. And we found that there were high frequencies of a one allele detected, and that's the P45S, which is the same as homologous to the P49S in Rhizopertha dominica. And in Australia, that's actually the strongest resistance allele that we have by phenotype testing. So they've selected multiple times for their strongest alleles. And those alleles are found across the country. Like, we've just published on southern India, but we've got data from northern India and around the country that they're the same alleles across the country. There are some areas where they're independently arising, but it's still the same allele. So we have data suggesting that it's probably the strongest allele. And they have multiple dosing and a lot of movement of their grain, but they only fumigate under bag stacks. So it's probably a similar situation to bunker-type storages. But anyway, so now we have a genetic test for phosphine resistance. We're now trying to implement them in using resistance monitoring, and especially in areas where the resistance frequencies are very low. They've only just detected some strong resistance in Western Australia, so we have an opportunity to try and detect where the problem areas are over there. And in active resistance management, so we can clearly and accurately assess the effectiveness of our strategies. We can look at reinfestation rates and whether resistance is moving back into a storage, with insect ecology informing us of where and when those interventions might be most effective. And also, with Paul, we're looking at synergists of phosphine because we know a lot of the biochemical basis, and so we're looking at the synergists that affect those pathways along with phosphine. and the effectiveness of alternative fumigants, etc. So, whoop. Yeah, and it's a very large team, but anyway.
Again, great story, but there's time for one question while we swap over.
Do you know why the resistance is much less in Western Australia than Eastern Australia?
Yeah, in Western Australia they invested heavily in phosphine from an early stage because phosphine is residue free and nearly 100% of their market is export. They said, okay, we're only going to use phosphine. They don't use any other kinds of insecticides or protectants on their grain. And so it's a cooperative, their bulk handle is the cooperative bulk handling, which a lot of farmers have shares in. And so they invested heavily into making their storages very sealable. And they also invested a lot of money into trying to make sure that their dose rates were very good. Yeah. And so the resistances they've detected are only on farms in fairly marginal areas from what I gather. And they just had multiple dosing on those farm silos. So, yeah.
Okay, thanks very much. Okay, so next we move on to David, who's going to tell us about weeds and their management.
Thank you very much. I think initially when this talk was given, I didn't know much, but in the morning, all those 2 sessions, I thought that, oh, my talk will be crap, There is nothing like that one. But anyway, if I see, you will see some of these management options. I joined this UQ last year, so I don't have mostly results, but what I try to do, like there are research needs in the weed area, what I feel, and when some people may not agree with all these ideas, that's why I take totally responsibility for this. Just for non-weeds people, little bit in a background, we have now conservation agriculture or conservation tillage systems, and that's because of that there are several advantages we know, social and economical and environmental benefits, but we have not that options of tillage to manage weeds. And what happened then, there are weed species shifts towards problematic weeds. Small seeded weeds, they're becoming a big problem. And for your information, weeds cost Australia around $4 billion Australian dollar. That's in control as well as in losses. If you see increased reliance on herbicides for weed management, it has increased herbicide-resistant weeds. We are somewhere, here, that's Australia, so that keep increasing in resistance cases. If you see glyphosate, I think in the morning we heard that there are 10 weed species in Australia already resistant to glyphosate. And I tried to put some of them. These are all our problem. There's Suncus, Bracharia, and these all from Australia. They are resistant to glyphosate. And once weeds are resistant to glyphosate, we have lost that important tool. Okay, so adoption of herbicide-tolerant crops is increasing worldwide. And I think we know because that's provide very flexible option of allowing this over-the-top application of non-selective herbicides. So what's happening now, we have got herbicide-resistant weeds. They keep increasing. In addition, like we have weed infestation, weed behavior that frequently change because of now management practices that changes, climate change, and ecological shifts. So therefore, I think existing management options, they need to be modified or improved. Okay? And there is a need, now a strong need to promote sustainable weed management strategies based on IWM principles. Just for your information, some of these problematic weeds, wild radish, most problem in Western Australia, annual ryegrass, wild oats, and these are some of our weeds in the northern region. That's windmill grass, fleabane, barnyard grass, feathertop, which is the most problematic weed at the moment, Eurocloa panicoides, liver seed grass, and then south thistle. These are some of the problematic weeds. Now, I think there is nothing new in this one. These are just a summary of different weed management strategies. I will not discuss each of them, but I will see where there is a need. For example, crop competition. I will discuss a little bit about rows, narrow rows, and also weed competitive varieties. weed seed destruction, I think Michael Walls will touch later on. Use of cover crops, targeted tillage. I think I will, okay, herbicide use. I will not discuss in this presentation, but I think there are a few good examples like about new molecules recently that GRDC and Bayer, and they had that agreement, and also another one is new uses of existing herbicides, double knock. I will discuss a little bit about them. Okay, managing weeds using crop competition. This is my favorite area. I want to manage weeds using crops. Okay, so using crop or agronomic approaches. Narrow rows, I think we know that one. This suppresses weeds more effectively than wide rows. Concept, as I mentioned, this is not new, but we need to do in this one in a more practical and economical way. Researchers now, for example, in Queensland, they are studying agronomy of legumes, for example, chickpea, and in the new GRDC project, a different row spacing. But this is all totally in weed-free conditions. What they found that increasing row spacing, that decrease yield of chickpea even in dry season. Okay, when we are talking about sometimes there are issue of moisture, soil moisture, but what they found that even in dry seasons, but most of these trials, they are in weed-free situations. So what I'm trying, now I'm trying to, need to collaborate with these agronomists rather than doing something separately or in a different project. So here I need to collaborate with the agronomist and do that agronomy, because later on when we say that narrow row spacing suppresses weeds more effectively, but there are some issues. For example, some people, or even some of these canopy, they may increase disease, some kind of disease. So I think what I'm saying that rather than doing something separately, there is a need to collaborate with these guys and do this agronomy at the same time. So recently we started some work at UQ, at the Gatton campus, and we found that in chickpea, for example, 25 centimeter rows, this suppresses weeds more effectively than 50 and 75 centimeter. Similarly, in mung bean also, similar results were found. So I think this narrow row spacing is very important. The second question, I think that's remained there, what its practical applicability. We will see later on. Another, it's important just for research at the moment, plant geometry. I think I have come from International Rice Research Institute, where recently we started some work on mechanical transplanted rice using this type of, and we had some discussion with physiologists. At the moment, like for example, maize or sorghum, we are growing in solid 1 meter row. But recently, last year, I found like near Dalby area, some farmers, they have got precision planter, and they're using paired row, and what they mentioned that they suppresses weeds more effectively. We didn't know, but recently, one agronomist at UQ, he also started something in sorghum and maize, and that's what I have that interest here, going with them and collaborate with them, whether they can suppress. This one I found in rice, definitely, for some varieties. And there are always some issue of it's about light capture issues. But still, I think we need to do, I call it like triangular planting. Some people, they call it diamond. So there is a need to do this type of research to manage weeds. Okay, weed competitive cultivars, very important component in the future farming, but not much research has been done because there is always trade-off between yield and weed competitiveness. Whenever we find a good or very weed competitive variety or cultivar, mostly its yield is low, and farmers don't accept that one. So there is a need to first, I think we need to consider first grain yield, and then try to find those traits, whether it is leaf area or biomass or height. Okay, and most important, this one, most studies at the moment, when we are talking about weed competitive varieties, they are focused on above ground parts, either it is shoot biomass or leaf area or tiller number. I think in our environment, definitely there is a need to include root traits. And when I say root traits, it's mostly because of water and nutrient limited environments. To me, I think mostly when we say weeds competing, that's mainly creating shade on crops. Also, they're taking their nutrients or water. So I think there's a need to screen those type of varieties for shade tolerance, high nutrient uptake, and water use efficiency. And definitely, I think there is a need to integrate this type of research with herbicide use, because I have seen most of these studies, when we deal or when we conduct, mostly in completely weedy or completely weed-free conditions. That we will never get in on farmers' field. I think we need to integrate with something like pre-emergence herbicides. So that's why, to me, I think the role of weed competitive varieties is not at the start. It's mainly after around 3 to 4 weeks after planting. So initially, you're assuming you have pre-emergence herbicides that works very well. Later on, that lose its efficiency. And then there should be a role of that cultivar. And to me, even if they are providing 10% suppression, I'm more than happy. You don't expect 100% from one component. So need to integrate or need to bring these different components, even if they are providing only 10%. Okay, another allelopathic cultivar need to evaluate. I think I was never interested in this area, but last 1 year now, I think I should do something on this allelopathic varieties. But I think there is a need, again, to integrate with herbicide dose and whether there's work or not. And mostly we are trying to get these traits from improved varieties. No, I think we need to go to wild plants and try to get that one. I will not talk much about herbicide tolerance, but I think there is a scope to screen these type of varieties also. Again, before that, we should have risk management strategies and stewardship guidelines to prevent. And I think most importantly, when we're talking about weed competitive cultivars, there is a need to collaborate with breeders. Okay? Without that, I think we can't get success. So I think, for example, Cement or Icrisate, they have got all this germplasm, bring that one and try to put in our local varieties. Some people, I think, as I mentioned, may not agree with that one, so I'm bringing a special, I'm editor-in-chief with crop protection. I'm bringing a special issue very soon, and that will talk about weed management using crop competition from all different countries and all different crops. So I think we will try to see whether there is a potential there. Can we manage weeds using crop competition? Okay, in our environment, second most important to me is fellow-focused weed control programs. Because we have got, like, in the northern region I'm talking about, summer rainfall, and that increases weeds problem. So soil moisture for us is an important issue, and I think if there are weeds there in fallow situation, so weeds may deplete most of that one. In addition, they also harbor pest, nematodes, for example, barnyard grass. It has got some kind of nematodes. So weed control in the fallow phase should be given importance. And something like including residual herbicides that would control number of cohorts rather than just relying on post-emergence herbicides. The double knock strategy, if people they don't know, double knock means using 2 herbicides, sequential application of 2 herbicides. And that's very common now in the northern region. So glyphosate followed by paraquat, that has been found to delay resistance. Also, I think important thing is cover crops. I think this is time when we have more herbicide-resistant weeds. We need to see, is there any applicability of cover crops? And I think recently Jeff Worth started some work in cotton, and I think it will be a good tool if we can find, especially for Roundup Ready cotton. And I think I will not talk much about these ones, but these are already in the field. Weed detection technologies. Targeted tillage, we recently started one project with Mike Walls, who is here. Robotic weeders, they have got potential, and I think to me this is great examples of collaboration between weed scientist and engineer. Okay, I didn't know, I initially means only last year I found that there is something called nanoherbicides, and I'm now interested in this one, and that's because we know that herbicide efficacy that's reduced because of several reasons, but some of them may be reduced uptake by weeds or through the applied formulation being unstable or rapidly degraded. So efficacy can be improved through the use of nanotechnologies. Nanoherbicides, these are usually 1 to 100 nanometer in particle size. So it means very small particle, and that means it's more surface area. So that reduction in particle size that increases surface area, and that results in increasing efficacy and uptake by plant and solubility in the spray tank. Some work is already going on this one, and I can't discuss that much here. Therefore, the use of these herbicides, they can manage weeds in modern agriculture without leaving toxic residue. That's also one of that important. When we say nanoherbicides, using less chemicals, less amount. Okay, and that could prove, I think it's very important in CA system to me, because we have got crop residue in the field, and that can absorb most of these herbicides. So these type of nanoherbicides, they can be very helpful in those systems. And they may also control that those weeds that have become resistant to conventional herbicides. Okay, bioherbicides, I'm not a great fan of this one, but recently, I think in the last 1 month, I discussed with a few people from the US as well as at UQ also. They are at the moment, I think they are used mainly in, mainly on roadsides or somewhere, but not in crops. So they are bioherbicides. Biotechnology, it has got a pivotal role, very important role in development and modification of bioherbicides. Now we know that climate is changing, evolution of herbicide resistance in weeds and lagging science. I think we need to try or something or evaluate efficacy of bioherbicides on crop weeds. And main risk that I was thinking that, okay, we don't want to spread any pathogen, we have not controlled, but we don't want to spread any new pathogens, but then I was told that these are very specific, okay? So there is a potential for use, but I think we need to combine with bioherbicides, with low doses of herbicides that may weaken weeds, and then it's very easy to control. I will not talk, I think we don't have enough time, but Mike Walsh will talk more about harvest weed seed control. That's also another important strategy, and I think it's about, just in simple words, destructing those weed seeds at harvest, whatever, and there are different systems, Harrington seed destructor or windrow burning. I think these systems, when we use with the herbicides, I think they can provide better control of weeds. Okay, and I think working well in the West, but need to evaluate its applicability in different regions. So recently we started a project with Mike Walsh, and that's in New GRDC, and we'll see on its applicability in our region. Okay, I think there is nothing new in this one. All people knows rotations of anything. I think that's the best thing. Rotation of your management, rotation of weeds, herbicides, rotation of crops, herbicides or tillage system. Even to me, no-till system also should be rotated, not after every year, but maybe after a few years. So I think we need to revisit this type of area. I have only 2-3 more slides. IWM strategy, I think this is a prerequisite to sustain available herbicide options. I know that none of these non-chemical approaches, they have potential to replace chemical weed control, but I think we need to have a kind of IWM approach. So need to integrate as many strategies as possible. I think our main focus in weed management should be to reduce weed seed bank before crop sowing and reduce weed emergence and weed growth in crops. And improved weed management should focus on shifting the crop weed balance in the favor of crop. I think there were a few challenges and opportunities. I think just, can I go on the last one? One is like convincing growers. I think I get like very excited when I say that, okay, this narrow row spacing can suppress. But it's very hard, to convince growers or researchers. So we need good communicators. That's also very important. It's not a good researcher also. Economics is very important. We can say, oh, use these 4 methods, but are they economic? So we need someone who can tell, yes, you can use these type of strategies. I think planter adjustment, it's very important. Means when we say narrow row spacing, is it applicable or not? Moisture issue, when we are talking about cover crops, do we have enough moisture? And I think need more farmers participatory research and need some kind of collaboration. I think that's very important for me, needs collaboration. Just last slide, giving my experience at Erie, we started some work. As a weed scientist, I was alone, did some work and had to work for like 5 years to get one kind of result. And that was not that efficient. Later on, we brought a strategy where I think we had a team. We took like 4-hectare research area, all public-private partnership, worked with these companies, and we had all different people in the same experiment. That's one even on nematodes, varieties, physiology, and I think same type of results we could get only in 2 years. So to me, I think this collaboration is very important, more important than our research. And I think more detail you can find in these 2 publications. With that, thank you very much.
Okay. A very quick question while we change over. If Michael comes to the front, please.
Thank you.
Okay, great. last but not least, we have Michael telling us about zero weed tolerance. Michael, take it away.
Okay, thanks, Ari. Yeah, so zero weed tolerance, I think that's the era that we're in now. We're looking to get our weed populations down to the lowest possible level, and it's all about increasing productivity. So this is a graph produced from some data by a colleague of ours at AARI, Peter Newman. It's a focus paddock study, so it's commercial fields where Peter has been monitoring the population of annual ryegrass in these fields over a 15-year period now. And the top line there is the one I guess I want you to focus on for the moment. It shows what you can achieve with herbicides. Herbicides are fantastic, we all know that, and they allow us to get our weed populations down to very low levels. And I'll talk about the lower line in a minute, but the lower line indicates that we can take that level of population density down to pretty much zero. So why do we need zero weed tolerance? Well, that top line in that previous graph averaged around about 5 plants per metre squared at maturity. Okay, so that works out on a roughly average paddock size of 200 hectares to be about 10 million plants in that paddock. So those plants, those 10 million plants that are there at maturity have survived herbicide treatments, and each of those 10 million plants is going to produce on average around about 200 seed. So what that means is that there's an enormous amount of genetic diversity that's going to be there at the end of the growing season, going into the seed bank, that will then subsequently cause problems in subsequent cropping phases. So this is an instance, Steve, where diversity is not such a good thing. So when you combine genetic diversity, cross-pollinated annual weed species, high population densities, it means you've got the potential for resistance to everything, not just herbicides, not just cultivation, fire, harvest weed seed control, but the whole lot, the whole gamut. So the message really is simple. You've got to get rid of that genetic diversity, and I guess the way that we're looking at it is that the only way feasible at the moment is to get rid of the population density. So getting back to my favourite graph, the bottom line, as I said before, shows that you can achieve zero. And that means you can really do a number on genetic diversity by getting those weeds out of your production system. So in this particular instance, in this study, the way that they have achieved zero, these farmers in commercial fields, have taken situations where they had in excess of 100 plants per metre squared setting seed at maturity down to 0 plants setting seed at maturity. They used herbicides, and then they used something called harvest weed seed control as well. So the top line, just herbicides alone. The bottom line, herbicides plus harvest weed seed control. Now the other interesting fact is that the guys who used harvest weed seed control, they didn't use it every year. On average, they were using it in 1 in 3 years. So that, I guess, demonstrates the impact of using an additional weed control tool on a relatively frequent basis. So the reason why harvest weed seed control can be effective is because of weed seed retention at crop maturity. So annual ryegrass, which is the focus of the previous graph, 88% of its total seed production retained at a height that allows that seed to be collected during the harvest operation. For wild radish, it's 99%. Brome grass, wild oats, a little bit less, about 73% to 85%. So high proportions of total seed production for each of those species can potentially be targeted by these harvest weed seed control systems. And we have a number of them available now, and these are some of the ones that have been around for some time. There are some additional ones, all very effective when they're allowed to work properly. They all focus on the chaff fraction. The chaff fraction is where the weed seeds exit the harvester. So if you get it right and you do a number on that chaff fraction, then they will be all similarly effective. Okay, so what has happened with the adoption of harvest weed seed control? Well, over probably the last 7 or 8 years, that adoption has skyrocketed. as people have become more comfortable with the idea of doing something at harvest, they have looked over the fence and watched their neighbours adopt these systems and use them efficiently to get their numbers down. We're starting to see high levels of high frequencies of adoption occurring right across the country. The top lines are for Western Australia predominantly. That's where a lot of these systems originated. But now that message is starting to be spread across the country, and we're starting to see some significant levels of adoption in the eastern states. Okay, so back to my favourite graph. It's not quite zero. I mean, everybody can see that along that bottom axis there, the line does bobble around a little bit, so it's not zero right across the production area. It's probably zero in a major proportion of the production area, but there are still weeds in the system. And as any weed biologist will probably tell you, it's impossible to achieve, or nearly impossible to achieve, eradication. But I think the message really is it's about the mentality that you take towards the situation. You don't just stop at 5 plants per metre squared where you've got the potential for problems in the future. You take those extra steps to keep driving those numbers down. And that means you have to start thinking about site-specific weed management. So low numbers are not enough. They cannot be tolerated in a paddock. So in this instance, we've got wild radish plants scattered across the landscape, maybe 1 plant per 10 square metres or less. We still need to come up with systems that can allow us to target those populations. And the beauty of site-specific weed management is that we can actually introduce new weed control strategies to target those individuals. The benefits are considerable. The farmers want this to occur. They want to get their numbers down even lower than what we're seeing here. Financially, it doesn't make sense for them to keep spraying whole fields to target low-density individuals. So, as I said, the benefits are considerable. There is a need for mapping and identification, and that's coming along. We will have some very good systems, hopefully in the not-too-distant future. Once we have that, then we can use a whole host of different weed control strategies. As well as alternate herbicides, we can go back to specific tillage. We can even introduce hand weeding, new technologies such as microwaving, electrocution, etc. These options all become feasible when we have that mapping and identification technology. So the value of a zero weeds approach for growers brings back flexibility into their production system. The weeds don't tell them what crops they can grow and when. They don't tell them when they can rotate and when they can't. There's timeliness of operations. They don't have to wait for emergence and germination before they can plant their crops, etc. As well as conservation of nutrients and just taking the competitive effects of weeds out of the equation. So there is demand within the industry to come up with systems that do lead to a zero weeds approach. And with that, I think I'll leave it there.
Okay, thanks very much, Michael. We've got time for a couple of questions, and we can perhaps get all the speakers to come up and have a bit of general discussion. So firstly, some questions related specifically to Michael.
Michael, I was interested in, sorry, Greg Wells at the AARM Sciences. I was interested in your picture of the adoption in northern WA. Is the key factor there, it's a two-part question, is the key factor there being forced to adopt new technologies? And if it is, what's your comment about, that Australian line?
Necessity is the mother of invention. Definitely herbicide resistance has played a huge factor in the adoption of harvest weed seed control systems. There's no doubt about that. Growers were forced to do something to avoid going out of business, and so absolutely. But I guess sort of moving on from that now, the opportunities for growers in other parts of Australia to avoid herbicide resistance and the loss of herbicides by adopting some of these systems earlier. That's what we hope is happening now.
Yeah, Michael, just a quick question. Did you observe, because last week I went out and tried to collect wild radish seeds in canola, but we had all the storms coming in a couple of days ago, and I found the majority of radish seed was all gone. Is it similar in Western Australia?
Well, and when the results from our survey across the West Australian wheat belt show that pretty much all the wild radish was retained above harvest height at the first opportunity to harvest, so right at crop maturity.
I mean, like the timing of rainfall in relation to the maturity might have an impact.
Yeah, there's no doubt that there'll be environmental factors that influence weed seed retention. Absolutely, there will be instances where harvest is delayed, wind, rain events will affect seed retention, for sure.
Yeah, really, that shocked me. I was going to collect some seed, this was all wrong.
I can get you some seed if you like.
Perhaps we can get all the speakers covered and continue our discussion. Deborah, Michael, Joe? Who am I missing? Great. Okay, so we're opening up for general discussion now, and obviously we've had a wide range of talks here. We go all the way from having quite theoretical models, such as Joe's, to something that's a bit more applied, such as Michael's. And then, of course, at the end, we have very strong applications in the sense of this last talk of Michael's is obviously talking very specifically about what you might do. And, of course, you have these wonderful adoption rates. I mean, amazing graph, I think. The adoption rate graph is actually more amazing to me than the graph that we're talking about. I wish we had an IPM graph like that, by the way. But over the general discussion, particularly want to hear from industry people. What are we missing that we really that we need to move forward in this area, in the management and the theoretical model area? And there's the sort of things that Joe's talking about, Michael's talking about, really useful. I mean, obviously we had a situation in the grains pests and the storage situations where we seem to have solved everything. So is there anything else that's needed there? So it's open to discussion.
Because, I quite like the method. Yeah, but thanks for you mentioned about the leaping. Sometimes we got investment to be short term and long term. But what I did a lot of leaping on wheat and canola, and we found sometimes it be difficult because we tried to use a leaping crop, and we had to do with breeding, and it's going to be a long-term process. And I don't think a lot of, in many cases, the funding body would happy to invest in a longer-term project. So,
Are you actually going to respond to that?
So I mean, sometimes it's not easy to find funding for a long-term project for about maybe 8 years because you need a broad breeding process.
I totally agree. I think that's why we need to have or collaborate with breeders and try to do, like, for example, okay, at Erie, when I was there, on weeds nobody is funding, mostly it comes from Bill Gates and all those things. But we later on we developed a project with breeders, how its applicability in terms of breeding. And so I think in allelopathy also, I think there is a possibility, but we need to have some kind of collaboration with breeders. And for me, I think, you need to have some like ARC type of grants, and there's a practical applicability there.
Yeah, just thinking about how funding body, I'm not just talking about GRDC, incorporate a short-term, long-term perspectives.
So is that something of concern to other people, a long-term perspective in terms of funding and support? Is that something we need to worry about? Comment from the back? Or a question from the back?
A question for Michael. We don't do much.
Which Michael?
Right, this is suppose long-term funding is related. We don't do much seed collection and harvest in the east. We're still using herbicide very much today, spraying seed, not seeds in that way. How can we spend long-term funding without actually getting out there and showing people how to do it, getting something that actually works? So it's economical. So it's just too difficult. So they just said it's too difficult. So it's frustrating. So how do you convince people and spend on something that's difficult to make money out of?
Yeah, well, that's the million-dollar question, isn't it? I guess the adoption that I've shown there has been driven by farmers. A lot of that adoption has occurred because we take farmers to go and talk to other farmers. So farmers who've used these systems, adopted them, and have realised the results, just go and tell their story to an audience of farmers, and the message is clear and the message is simple, and we see adoption occurring pretty much straight away. That's the approach we've taken. I mean, you can't force people to want to change their practices, but if it's an obvious thing you can try, and it is fairly simple and fairly easy, listen to other farmers who've done it.
Next step.
Work that you put up in your work, there was also a grazing component to those rotations. Was there about trying to reduce ryegrass numbers through pasture growth?
There was some. I mean, it's not a big area for grazing. It's the northern region of West Australia. There's not a lot of livestock there anymore. So if there were pasture components that was there to try and get the weed numbers down, and certainly that was an option that was practiced in some instances at the beginning.
It's certainly gone out of a lot of areas, broadacre farm, sheep and so on.
Yeah.
Even a bit north of SA.
Yeah. It's as many crops as possible.
I have a question for Joe. So it's very interesting your talk about low dose, high dose question, and it's one of those ones where I still don't think there's a really clear answer, to be honest with you. So one of the strategies we've tried in cotton is for some of the pests where most control options are fairly broad spectrum, one option to increase activity is to reduce dose. So if you reduce dose to a point where you're still getting sufficient control, some of the beneficials of some of them, we're getting the additional biocontrol from those after the spray. So is that something you can factor into your model to see that?
It is, yeah, I've not done it, but it's certainly testable. Yeah.
There's a lot of complexities to that, so it's a simple question, but there's a lot to it. When you go around, I want to know the answer.
And of course, dose isn't constant data because, of course, it changes over time. Any other points people want to raise? Yeah.
I have one question. In Australia, for weed control, Australia already use very low rates compared to other parts of the world. I was wondering whether for fungus, insect control, Australia use low or high rates compared to other parts of the world.
Anyone want to go downstream that? Are we higher or lower in terms of fungicide use?
I could probably make a comment. In terms of fungicides, sometimes we're equivalent, and in many cases we're lower in other parts of the world.